It's very not that it's not easy.
Okay, we're going to go ahead and get started here, and we're going to continue on with metabolism. Probably going to finish this chapter today, and that would take care of Unit 2. So we'd have cells and metabolism taken care of. So that means we're going to have a test at some point. And I'm thinking, let's see. No, it will not be immediately after we come back from spring break. So we can decide that when we get back. So the earliest would be if I open up the bone test on the Wednesday after spring break, is after we're back from spring break. I open up the bone test, that would give you a couple days to get ready for the bone test. Then we could have the lecture test the following week. We'll talk more about that as we get closer. So anyway, we're going to wrap up metabolism. Anybody have any questions so far? Okay, and I'm going to go into this, and I think this is where, here we go, metabolism. So metabolic activities inside the cell. Our metabolism is the sum of all of the chemical reactions in our bodies. Tearing down proteins, building up proteins, tearing down sugars, building up glycogen and stuff like that. Our metabolic activity basically consists of two types of chemical reactions. We either make large molecules from small ones, and that's called anabolism, or an anabolic reaction, where we take something like amino acids and we make a protein. Or we have catabolic reactions where we take large molecules and break them down. We break them down into small molecules, like digesting starch to glucose. Anabolic reactions, anabolism lets us make the proteins we need for growth and repair, makes the proteins we need for antibody production, to make more muscle, to make our muscle cells larger, to make our bones stronger. We take all the foods that we eat, and we can link them into larger and larger structures. We can store sugars as glycogen. We can store, we can take amino acids and make proteins, not store them, but we can turn them into proteins. So, because that's what a protein is, a long chain of amino acids linked together. So these are anabolic reactions. They always require energy. It takes energy for an anabolic reaction to occur. Catabolic reactions are just the opposite. Catabolic reactions release energy. Catabolic reactions break large molecules down into small ones. We break apart the carbohydrates we eat. We break apart the fats that we eat. We break apart proteins and other amino acids so that we can reuse those amino acids. And so a catabolic reaction will always release energy. And we use that energy then to build up other molecules. We use the energy from catabolic reactions to do things as well as build up other molecules. So the key thing about metabolic activities is that we want to use the energy found in food. We want to use the energy found in food and transfer it to the structures of our bodies, the cells of our bodies, to do things. We want to be able to do things. We want to be able to walk and talk and breathe and eat and write things down and read and drive. And we want to be able to pump blood and inflate our lungs. We want to enlarge our muscles. We want to rebuild our bones as necessary. And that all takes energy. And we can't release the energy directly from food into our bodies. We can't release the energy from sugar all at once into our bodies. It would be, it's too much energy to release at once. We have to release it slowly and gradually. And the way we do that is we take the energy from glucose and we park it temporarily in ATP. ATP is is the currency, the money of our bodies that we use to transfer energy around to the cells where it's needed. We can't just break apart a molecule of glucose and say, hey, here's a whole lot of energy, do something with it, go and do good. It doesn't work that way. We have to transfer it gradually. Our bodies couldn't process all that energy being released at once. Imagine if all the energy in your gas tank, was suddenly released into your engine at one time. It would be very exciting for a very short period of time. But then what would you do? We can't release all that. We have stored energy in the gas tank. We have potential energy in the gas tank. You can't turn all that into kinetic energy at one time. We have to release it slowly. And that's what we do with our food. We can't take all the potential energy in a potato or a Snickers bar and release it as one at one time. We could, because we would waste it. We would not be able to process the energy that we need. So, and the way we do this, the way that we can distribute our energy around so that we can use it when we need it and not waste it, is through ATP, adenosine triphosphate. The third phosphate on ATP, an ATP molecule has 3 phosphates attached to it. And it is this third phosphate that held together, number 3 position. Let's erase that scribble down here. Okay, now. Okay, so stop. Just leave it the way it is. So we have the third phosphate, you see. The third phosphate there is held onto the other 2 phosphates. This is phosphate number 1, this is phosphate number 2, this is phosphate number 3. This is a high energy bond. When we break that bond, release energy. When we break that bond, we release energy. When we put the bond back together again, it requires energy. We use ATP to go around. ATP will break apart into ADP plus inorganic phosphate. This is inorganic phosphate. But it will also release energy. So ATP, for 7 seconds, it can carry that phosphate, and then within 7 seconds it has to release that phosphate so it can release energy. So in most of our chemical reactions that require energy, we will bring an ATP to the site, release energy, break bond, and release the phosphate. So, and the whole process of using ATP involves putting the third phosphate on or taking the third phosphate off. The term here that you need to remember, this is one of those words that you have to remember, is phosphorylation. Phosphorylation, adding the phosphate to a different compound, adding the phosphate. So here we have, if we have ATP and we break it down to ADP, the diphosphate, and the inorganic phosphate, the third phosphate with the energy, that goes to another molecule. That says molecule. And it picks up the energy. So the energy, that says energy. So I can't even bother fixing that. When we transfer the phosphate to another molecule, we bring the energy with it. And now to get our ADP, to get ADP again, now we have to apply energy, add energy, and we can go back and get ATP. So I've probably overdone that whole thing, but the point is that we use ATP as our currency to transfer energy around, to move energy around in our bodies. And it's that third phosphate that does all the work. You either release the third phosphate and provide energy to do something, or you force that third phosphate back on with energy so it can be reloaded and go do something again. So now, metabolic reactions are controlled by our enzymes. Enzymes lower, the classic definition, what does an enzyme do for us? Well, they lower the activation energy needed to start the reaction. Instead of having to spend a lot of energy to get the reaction going, it goes to the slower reaction. Imagine if you could get water to boil at 100 degrees Fahrenheit instead of 212. You'd have less energy required to get water to boil. That's what an enzyme does. It lowers the amount of energy necessary to get a reaction to go. You put a pan of water on the stove, you turn the heat on, and it takes a while to get the water hot enough to boil. It has to hit 212, 100 degrees Celsius or 212 degrees Fahrenheit. That's boiling point of water. But what if water boiled at 50 degrees Celsius or 100 degrees Fahrenheit? What if we had some magic ingredient that we could add to it to let it boil at a lower temperature? That's what the enzyme does. Enzymes lower the energy necessary to start a chemical reaction. The enzymes are not destroyed. The enzymes are used. They allow the reactions to occur. And the way they do this is simply by bringing the reactants together. If you have two chemicals that are going to react with each other, and they're drifting around aimlessly in a liter of water, they're sort of floating around in here. If there was some way to bring those two chemicals together faster than just random chance, they'd react faster. If you have a cake mix, if you put eggs and flour and sugar and oil and whatever else you put into a cake mix, cake batter, and put it into a bowl and set it aside. You notice I left out the part about mixing it. What if you didn't mix a cake batter? You didn't mix it up. You didn't put the beaters into it, the mixer into it, and run it at 3 minutes at at high speed or whatever. What if you just sort of threw it all together in the bowl and let it sit there? What's going to happen to the cake batter? You're not going to get cake batter, for one thing, because you're not going to get the chemical reactions needed to form the batter. You have to apply energy to that with the mixer. That and would you rather mix it by hand or use an electric mixer? Mixing it by hand takes time. Mixing it with the mixer takes a few seconds. So what an enzyme does, an enzyme allows a chemical reaction to proceed at a faster rate with less energy. Enzymes are specific. The chemicals that the enzyme brings together are called the substrates, and every enzyme has an active site on it. That's the place where the substrate, the chemical, can land, and that is unique. So there are enzymes for everything. And while we can't do anything about temperature or particle size or concentration, we can flood an area with enzymes to get things to happen. So we see the illustration down here. There's our enzyme. These are the substrates are the chemicals that you want to react, and they have an active site on the enzyme. When they come together on the enzyme, they react and give us a product. The enzyme lets go and it's ready to go again. So we depend on our enzymes for chemical reactions to work because we're dealing with very small concentrations in our body. We're dealing with small particle size, and we're stuck at 98.6. We're stuck at body temperature, 37 degrees Celsius. Okay. When we process food, three things are going to happen. These are the stages of metabolism. First of all, we're going to chew the food. We start the process of digestion in our mouth. We start chewing the food. We continue digestion in our stomach. Our stomach churns and swirls and starts breaking down proteins. It continues to break down carbohydrates. We start breaking down carbohydrates in our mouth. We get into the small intestine. We finish the process of breaking down protein, and now we move into the rest of our small intestine. The first 10 inches of our small intestine is where we break down our proteins and fats, and we start absorbing nutrients in the rest of our small intestine, the remaining 19 feet of it. Once we absorb these nutrients, we transport them to the liver for cleaning, up to the heart for distribution, and then we take all those nutrients out to all the various cells. And that's the first step. It's a pretty big step in here. We've only just gotten, at the end of the first step, we've finally gotten to the cells. You eat a banana, digestion starts in the mouth. We have to continue digestion in the stomach. We start absorbing in the small intestine and then transporting to the cells after we go through the liver to clean it up, and then up to the heart to send it out through the systemic circulation. So now the nutrients have gotten to the cell, to the cytoplasm. Once the nutrients get to the cell, we can make fats, we can make proteins, we can store carbohydrates as glycogen. So we can make all these large molecules. We can have anabolic reactions going on. We can take amino acids and make proteins. We can take the various fatty acids and glycerol and we can make fats. We can take glucose and store it in long, long chains called glycogen. Or that all takes place in the cytoplasm of the cell. Or we can take glucose and break it down through catabolic reactions, metabolism. We will break glucose down in a process known as glycolysis and release energy. So, we can, this is all in, back. Here we go. Once we get to the cell, we can, these are anabolic reactions here. Glycolysis is the breakdown of glucose. We release energy here. Glycolysis is the lysis of glucose. We can break it down into ultimately carbon dioxide and water and the release of energy. Now that's in the cytoplasm. The third step of metabolism takes place in the mitochondria. This is in the cytoplasm. Stage 2 is in the cytoplasm right here. Stage 3 takes place in the mitochondria of the cells. And we always end up with carbon dioxide, water, and the release of ATP. So let me, now that I've got you good and confused, let's go a little further and try and clarify all this. Now, if you're visual like I am, this is a much better way of showing it. Stage 1, digestion in the gut. Digestion starts in the mouth with starches. Carbohydrates start being broken down in the mouth with the enzyme amylase. Proteins are broken down in the stomach and in the small intestine to their amino acids. And fats are broken apart, chopped apart in the small intestine into glycerol and fatty acids. Now, so we've digested everything. Once we get everything through the liver to get cleaned and get cleaned up and goes out, now these nutrients go back out to the cells. Anabolic reactions occur. Anabolic take energy. We take amino acids and we make proteins. We take amino acids and we make proteins right here, long chains of proteins. We take glycerol and fatty acids here and we make fats and we store them in adipose tissue. And if we have lots of sugars, we can make glycogen. Excessive amounts of sugar end up being stored in our bodies as glycogen, usually in the liver, but also in muscle cells. Now, there is also, and this is all taking place in the cytoplasm, right here. But some of that glucose is going to get used to release a little energy. And that also takes place in the cytoplasm. And it's called glycolysis, right there, glycolysis, the lysis of glucose. And the third step takes place in the mitochondria, where we have what we call the actual oxidative breakdown. This step uses oxygen. This step, no oxygen. Glycolysis doesn't require oxygen. We don't need oxygen to get some energy there. The only place in our bodies where we actually use our oxygen is in the mitochondria. That's how we release the greatest amount of energy from our nutrients, from glucose, is through the use of oxygen. Let's back up a little bit, talk about this in more detail. We know what energy is. It's the ability to do something, to do work or change something, to move it. We respond to heat energy, light energy, sound, electrical, mechanical, and chemical energy. Our bodies are involved in all that. We generate body heat. We depend on light rays so we can see. We hear things, so we have sound waves, that's sound energy. Our bodies are electrical. We have a charge on our cells. We have mechanical energy. We do things, and we have chemical energy stored in adipose cells and as glycogen. And we're always changing energy from one form to another. Sound waves are converted to mechanical energy because they cause vibrations. And those vibrations are converted to electrical energy so that we can generate a nerve impulse for a sound. We take light rays, light energy, and convert it to electrical energy so that we can generate a nerve impulse in our eyes. We generate heat all the time. We use our muscles to pump blood, to move our bodies, to breathe, to squeeze food through our digestive system. That's mechanical energy. And we took chemical energy to do that. We took the energy from glucose to power our muscles. We took energy from glucose and lost some of it as heat energy. So we're always moving energy. We're always changing the forms of energy that we're dealing with. And every one of these are involved in all of our metabolic reactions. So chemical energy, the chemical energy that we depend on. So where do we get this energy? Well, the energy that we use in our bodies to do things, most of that is based on chemical energy in the form of glucose. We store glucose in the form of glycogen. We can store fats and convert them to glucose, and we use that energy. But most of our activities depend on chemical energy based off of glucose, and we oxidize the glucose. Oxidation, it's sort of like, it's almost like we're having a bonfire and we're burning the glucose to release energy. If you have a fire pit and you burn wood in the fire pit, you release heat and light energy. Heat and light energy become available because you're releasing the energy stored in the wood. And you can't do it without the presence of oxygen. Same with glucose. We oxidize our glucose in the presence of oxygen, and we release a lot of energy that we can use to run our metabolic activities. Cellular respiration is how we is a term we use to describe converting the energy stored in food, all that potential energy in the valence shells, in the valence electrons, how we can take that energy and use it in our bodies. So cellular respiration, 3 steps to cellular respiration: glycolysis, the Krebs cycle, and electron transport. And I know it says citric acid, but this is really known as the Krebs cycle. Okay. Glycolysis, Krebs cycle, and electron transport. Those are the 3 stages of cellular respiration. The end result of cellular respiration is going to be carbon dioxide. The carbon dioxide that we're breathing out right now comes from cellular respiration, and only one stage of cellular respiration. We release water. We produce ATP. Cellular respiration provides us with the energy to force the third phosphate onto ADP and park it there for 7 seconds. And we give off heat. About 60% of the energy that we consume in food is released from our bodies as heat. We only get to use about 40% of that energy to do things. So we have carbon dioxide, water, ATP, and heat energy being released. 2 terms here: anaerobic. Anaerobic means without oxygen. Anaerobic reactions will allow you to generate a little ATP, not much, just a little. Aerobic reactions with oxygen allows you to make a lot of ATP. Our metabolic reactions work better in the presence of oxygen. They'll still work without oxygen, but they work much, much better with oxygen. So now here's a term for us: oxidation-reduction reactions. When something is oxidized, it gains oxygen or it loses a hydrogen. Now, that's confusing. Here's an easier way to consider this. When something is oxidized, it loses electrons. When it loses electrons, it loses energy. So when something is burning, for example, let me put this on the camera. Here, if I light this device, I'm burning the fuel inside the lighter. Okay, so we're seeing a flame, we're giving off heat, we're giving off light. This is oxidation of the fuel. It's now for that to work, to give off the heat and light, we have to be breaking chemical bonds and releasing electrons. So we have electrons going out here. They're being released. Oxidation is the release of electrons and the release of energy. When we burn something, we are oxidizing it. Now, where do those electrons go? Well, those electrons have to have some place to go. And when we're doing this, the electrons are being grabbed by the oxygen in the air. So this is what's happening at this point. Now, let's go back. There was another term here I wanted to point out, and that is reduction. When something is reduced, so when something is oxidized, it loses electrons and releases energy. When something is reduced, it gains electrons and gains energy, because we can't have, electrons will not randomly go flying off by themselves. Something has to grab those electrons. So when something is burnt, something is burning, we are releasing electrons. But for the process of burning the fuel to continue, as those electrons come off, they got to be captured by something. And when I'm doing this, I'm breaking chemical bonds and releasing electrons. The only reason that it's burning is because oxygen is grabbing those electrons. So we use the term oxidation. The release of electrons, the release of energy. Where are the electrons going? Well, they have to go to oxygen. Oxygen needs more electrons because it's shy 2 in its valence shell. It has 6 electrons in its valence shell, and it really wants those 2 extra electrons. So every time we burn something, we release electrons, and they get captured by the oxygen. What if the oxygen wasn't present? Then it wouldn't burn. And so we say oxygen allows the reaction to occur, but we also say the substance that grabs the electrons is reduced. The oxygen grabs those electrons, oxygen gets reduced. Now, I know it makes absolutely no sense to say reduced if it's grabbing more electrons. Chemists will tell you that when a substance is reduced, it has become more negative because it's grabbing electrons. I didn't understand that very well when I was an undergraduate, and I still don't understand. Now I don't understand why they say it that way, but all you have to remember is that oxidized substances, when something is oxidized, it gives off electrons and gives off energy. When something is reduced, it grabs those electrons and grabs energy. So now, we call these chemical reactions where we have oxidation and reduction going on a redox reaction. Something is going to give up the electrons. We call that the electron donor, and it is the reducing agent. Again, it doesn't make any sense. Just take it for face value. The electron acceptor is called the oxidizing agent. Now think about that for a second. The electron acceptor is going to be the oxidizing agent. And if you consider that this is usually oxygen, you're burning a candle. That's a candle, by the way. There's the wick, and that's the flame. You're burning a candle. You're giving off electrons. Those are electrons. Something has to grab those electrons. It's usually going to be oxygen. Oxidation can't occur without oxygen. So we say oxygen, the electron acceptor is grabbing those electrons. It is the oxidizing agent. No oxygen, no flame. Now, the paraffin is the electron donor. It's giving off the electrons as we break the valence bonds here in the candle wax. That's the electron donor, or the reducing agent. But it's not going to work unless you have the oxidizing agent, which in this case is oxygen. It's nearly always going to be oxygen. Now, let's see if I can make this any better for you. So, okay. Now, this is a incredibly confusing picture. I'll just tell you that. But if you trace the line, like look at compound A to the upper left, it has 2 electrons. It is the reducing agent. It has electrons it wants to give away. If something allows it to give off those electrons, compound A will become oxidized and those electrons are gone. But something has to grab it. In this case, compound B, which is oxygen, grabs those electrons. And compound B is now reduced. To have a successful flame burning, the oxygen has to become reduced by grabbing those electrons. Now, in our cells, we have redox reactions taking place all the time. We transfer energy by using a phosphate. When we oxidize something, it's losing electrons. When we grab electrons, we're reduced. And we have to be able to transfer that energy. The energy coming off can only be transferred by using phosphate. Now, this last bullet here, this is chemist speak. When you add electrons to a compound, it reduces the number of positive charges. That's how we get reduction. Makes perfect sense to me, I guess. Reduction, we add more electrons, we become more negative, we reduce the number of positive charges. Okay, now, this is an oxidation-reduction reaction right here. Oxygen gets consumed, carbon dioxide and water are given off as the electrons are released. Now I want to show you a little demonstration here. Hopefully this will work. I can show you the release of electrons. Let's do the camera here. We spared no expense on this demonstration. But here we have a, let me set this up a little higher. Here we have a candle. You'll see it's a real candle. And I'm going to light the candle. And we got successful oxidation of the, let's show that. There we go. There you can see the candle then. Okay. So we got the candle burning. We've got successful oxidation of the wax here. You can see the flame, it's not a battery powered anything. So now, the reason it's working is because of the presence of oxygen. The electrons being released when we're breaking down the wax. What we're doing is we are breaking the bonds in the wax of the candle, and that's causing a release of energy. That's where the flame and heat and light are coming from. The electrons coming off of that wax are being grabbed by the oxygen. Now, let's say we take the oxygen away from it. Now, watch what happens here. Let's see if I can get this to come down to a little better. There we go. Now, there's our candle. And now notice the top of the glass container. The candle is out, but let me do this again. What I want you to look for is a fog or a film that forms on the surface of the glass. There's our flame burning, and let's get this down a little lower here for us. There we go. Now watch the surface of the glass when I cover it over and see what happens. Now the flame is going to get smaller and smaller and smaller. And if you look carefully here, perfect demonstration, I think. What's happening to the candle? The candle is going out. What else is going on here? If you note, it's sort of hard to see, but if you look carefully here on the edges of the glass, if I put a piece of paper in, see it right here. That's going to help or not. We're getting a fog in this area here. What am I trying to show? What I'm trying to show you is that as we consume the oxygen inside the container, we get a deposit of water vapor on the inside of this glass. This glass area here is going to get fogged over with water vapor, meaning that we're releasing water. The end result of cellular respiration, of oxidation reduction, is going to be carbon dioxide and water. Now, this one how do I know that we're generating carbon dioxide? And what's the common answer here that the candle goes out? The common answer is, well, you've used up all the oxygen. No, there's plenty of oxygen in here. What's really happening is that burning the wax causes the release of a lot of carbon dioxide. Carbon dioxide is heavier than atmospheric air, and it pushes the air away from the wick. If I try to light this, I can do it this way. This worked yesterday. The reason that the candle goes out is not because we've used up all the oxygen, but it's because we're displacing the carbon dioxide, and we're displacing the oxygen with carbon dioxide. The candle's going to go out here shortly. There it goes. And now, do I have enough oxygen inside there, or have I displaced it with carbon dioxide? I can't even, the match will go out as I get close there, and we get no flame. We've pushed all the oxygen out. Carbon dioxide occurs. And by the way, if you notice, when I held the match close to the glass, like this, you'll notice on the side of the glass, it got foggy. That was water vapor forming there in the side. So all these demonstrations here that actually work so much better in person than through cameras. All these demonstrations did was show that the end result is that when we are oxidizing something, oxygen grabs all those electrons. The end result is always going to be carbon dioxide and water and the release of energy. Now, our bodies control this. Our bodies do not release a lot of energy all at one time. We can't, because we would explode. We have to, the amount of energy in glucose is amazing. So we have to release that energy slowly. The amount of energy stored in that candle, we can't release all that at once. That would be a very explosive step. So even with that candle, we have to gradually break the valence electrons, break the covalent bonds, releasing those electrons, releasing the potential energy, releasing in the form of heat and light. In our bodies, the energy in a candy bar, for example, you get 180 calories in a Snickers bar. You don't want to release all 180 calories at one time. You have to release them gradually. And that's one of the problems we have with the simple sugars, like in a Snickers bar. We release a lot of energy at once. You know, if we release, we get too much energy released at one time. we burn through it quickly. Eating an apple, on the other hand, which may not be as tasty as a Snickers bar, maybe, but has complex carbs, allows us to burn through that slowly. Now, the process of cellular respiration is to break down glucose in a controlled series of chemical reactions. And what we're doing here is when we release electrons, we're actually going to release hydrogens with it. See, because hydrogens and electrons, come on. Here we go. Hydrogens have a positive charge. Electrons have a negative charge. If you want, whenever an electron leaves, like in the case of the burning candle, when an electron leaves the wax, when we're breaking those bonds in the wax, it's going to drag along with it its best friend in the whole universe, a hydrogen. So every hydrogen ion is going to be traveling with an electron. They stay balanced. So if we want to get electrons to go somewhere to be picked up by oxygen, we drag hydrogens with it. That's why we get water. The oxygens are going to grab electrons, and those electrons are bringing hydrogens. So the oxygen grabs, well, I need 2 electrons in my outer shell, so it grabs those 2 electrons, but the electrons brought their hydrogens, and all of a sudden we got a water molecule. That's why we get water vapor forming on the inside, condensing on the inside of our glass. Now, so we find chemically in our bodies, it's easier to move the hydrogens around than it is to move the electrons. The electrons are going to, what we want is the electrons to go with their energy. The hydrogens end up dragging the electrons with them. So in our bodies, we have a very controlled release. This is what happens if we were to have uncontrolled oxidation. Our bodies would likely explode. This is what would happen if all the energy, potential energy in your gas tank let go at once. This is what would happen if all the energy in that Snickers bar let go at one time. You know, this is a real-time video of the Hindenburg exploding in 1937 in Lakehurst, New Jersey. It killed 30 people out of 90 when it crashed trying to land, because the fuel tanks, the tanks, not the fuel tanks, but the gas bags that made the Zeppelin, the blimp essentially, lighter than air, were filled with hydrogen, not helium. So, okay. We don't want to do that to ourselves. So in chemical reactions in our bodies, to get electrons to come to oxygen, we got to have, or to be accepted by something, we end up dragging the hydrogens along with it. So we use coenzymes. Now, two coenzymes you got to remember. NAD plus is a coenzyme that we use. It is an electron acceptor. NAD plus, accepts hydrogens and automatically electrons. We also have another one called FAD, does the same thing, picks up hydrogen ions and electrons. This is how we can transfer energy around with inside the cell before we make the ATP. So remember those two terms, NAD plus and FAD. The names nicotinamide adenine dinucleotide. That's why we call it NAD plus. It's plus because it has a positive charge on it. FAD doesn't have a plus or a minus because it's neutral. Okay. Now, in cellular respiration, if we're going to use oxygen in our bodies, we call it aerobic cellular respiration. And most of the energy gain that our bodies get comes from aerobic cellular respiration. We use oxygen to strip away most of the electrons from glucose and most of that potential energy that comes off of here. We take the electrons off and the energy off of glucose with their hydrogens. Because when you take an electron, you have to take the hydrogen too. So if you take the electrons off of glucose, we transfer those electrons and the hydrogens to oxygen and we get water. We form water. So we produce water and we produce carbon dioxide and we release a lot of energy because all that potential energy that we're stripping off from the electrons is available to use. And the chemical reactions in aerobic cellular respiration are based on stripping off hydrogens and dragging their electrons with it. So, reminder, it's the valence electrons that have the potential energy. The valence electrons of the carbons and the hydrogens have lots of potential energy, and they transfer that to oxygen. Oxygen is the big electron acceptor, and it takes it causes energy to get released, to be released from those valence bonds. When I lit that candle and the wax was burning, I was pulling electrons away from the carbon and the hydrogens in the candle wax. The electrons and the hydrogens were being grabbed by oxygen, and we were releasing heat and light. Without oxygen, we're not going to get a flame. Without oxygen, we're not going to have oxidation, at least very much oxidation. Oxygen allows lots of electrons and hydrogens to be ripped away from a molecule. In the case of the candle, we're ripping the hydrogens and electrons away from the wax, carbons and hydrogens and oxygens already there. We're ripping the electrons and the hydrogens away from the wax and releasing that energy in the form of heat and light. As long as oxygen was there to grab those electrons, it would continue to burn. As soon as you took the oxygen away, no burning. We know that the electrons in the valence shells have the most potential energy. So those are the ones we go after. The valence electrons are the ones that have the most potential energy. So when we are oxidizing something, we're breaking the valence, we're breaking the covalent bonds for the valence to release the valence electrons, because they've got lots of potential energy. That's what we want. We couldn't burn a candle if we didn't have lots of potential energy. Now, this is the formula for cellular respiration. You'll probably want to commit this to memory. I'm sure that you'll treasure this always. I don't think no one has ever come up to me in the street and said, "Hey, what's the formula for cellular respiration?" Not going to happen. However, glucose in the presence of oxygen will give you 6 carbon dioxides plus water plus energy. So where does it, we breathe out carbon dioxide. We generate carbon dioxide during cellular respiration. We need to understand that in healthcare, that the cells are generating carbon dioxide. If we're generating an adequate amount of carbon dioxide, it's going to show up in our blood. And that's as a result of cellular respiration. The burning of glucose by oxygen releases carbon dioxide. It also releases water, and it releases energy. The energy that we use to do things in a large part depends on the presence of oxygen. And if it's working, we're going to be breathing out CO2. We give off carbon dioxide, we give off water, and we release energy that we use to make ATP. So C6H12O6 plus 6 oxygens yields 6 carbon dioxides and 6 waters, and the release of a lot of energy. So in healthcare, it's important to understand where the carbon dioxide is coming from, where the energy that our bodies are using is coming from. Okay. Now, our bodies are somewhat peculiar because we have an anaerobic process that doesn't use oxygen. Now, we say that oxygen is the ultimate electron acceptor, and we can't oxidize anything without the presence of oxygen. However, there is anaerobic cellular respiration too. It takes place in the cytoplasm, and it uses that NAD plus as the electron acceptor. It is NAD plus is an oxidizing agent. Normally, we think of oxygen as the oxidizing agent, but NAD plus is an electron acceptor, and that makes it an oxidizing agent. So you can strip off NAD plus and strip off hydrogen ions and electrons and oxidize something. Not very well, and not a lot of energy gets released, but you can still use NAD plus. Okay. Don't forget, electrons always take their BFFs, their hydrogens with them when they get moved around. When we are stripping electrons from glucose, stripping them off to release energy, when we strip off the electron, we're dragging a hydrogen with it. When we're burning a candle and we're stripping off electrons, hydrogens are coming with it. How do we know that? Because water vapor is condensing on the inside of the glass. That means that the electrons are dragging their hydrogens along, which are combining with the oxygen in the air to give us condensed water vapor. How do we know that carbon dioxide is being formed? Because the candle goes out. There is plenty of oxygen available inside the glass container, but we've generated so much CO2 so quickly that it displaces the available oxygen. This whole process is nothing but a transfer of energy. We transfer the energy from food, the high energy bonds in food, we ultimately end up transferring it to oxygen. We transfer electrons and hydrogen to oxygen. We start out with a Snickers bar or an apple or a potato or a steak or something, a piece of bread, whatever. Lots of high energy electrons in there. And as we strip it apart, those hydrogens and those electrons are transferred to oxygen, and we form water, and we're releasing the energy. Food in the glucose in the presence of NAD plus will release energy for us. So we park all this energy. Now, this is an important point here. We park all of our energy in the form of ATP when we're using it. When we release the energy from glucose, we form ATP. Every ATP molecule formed contains 7.3 kilocalories of energy. 7.3 kilocalories of energy. regardless of how we're going to get there, we're going to make 7.3 kilocalories of energy. There are two ways to make this. What we call substrate level phosphorylation. That takes place in the cytoplasm. It involves a process known as glycolysis. Substrate level, it uses enzymes. It uses enzymes for this to work. Oxidative phosphorylation takes place using electron transport. So you got to remember the two types of phosphorylation. Substrate level phosphorylation takes place with glycolysis in cytoplasm. Oxidative phosphorylation takes place in mitochondria. Big payoff. Little payoff. They both are going to give you ATP. You're going to get a lot of ATP from oxidative phosphorylation. You're going to get a little ATP from glycolysis. Okay. Now, substrate level phosphorylation uses enzymes to transfer phosphates from one molecule to another. The phosphates contain the energy. So substrate level phosphorylation uses enzymes. Substrate level phosphorylation takes place in glycolysis and in the Krebs cycle too. So we transfer energy from one product to another in the form of the phosphate. Substrate level phosphorylation, not a big payoff, only in the cytoplasm or in the Krebs cycle. Oxidative phosphorylation takes place inside the mitochondria. We transfer electrons. We're actually moving electrons around, and we get a big payoff of energy here. We produce a lot of ATP through oxidative phosphorylation. We use a process known as electron transport. Oxidative phosphorylation involves the movement of hydrogens across the membrane of the inner membrane of the mitochondria and allowing them to pass through a special pump that makes ATP. So this is called oxidative phosphorylation. High payoff, a lot of energy involved here. So we start out with glycolysis. Let's keep it simple. The oxidation of glucose gives you about 36 ATP, important number to remember. Different textbooks are going to say different numbers, but 36 seems to be a general number here. 36 ATP. And you multiply 36 times 7.3, it comes out to something like 260 or 280 kilocalories. 280 kilocalories of energy. That's what we get to use. That represents 40% of the available energy in a molecule of glucose. 60% is lost as heat, but 40% is what we get to use. 60% comes off as heat. And most of our body heat comes from our skeletal muscles. Now, glycolysis, the Krebs cycle, and electron transport. These are the three steps here. Oops, I didn't want to do that. All drawings. Now cut it off. Okay, now. This is an incredibly busy slide. It shows you the three steps of cellular respiration, starting with glycolysis and ending with electron transport. So I'm going to just, this is so busy that I never consider it useful in here. But the thing to remember is that glycolysis takes place in the cytoplasm. This is a mitochondria. This is inside the cell. But the glycolysis, it's floating in the cytoplasm. But the process of glycolysis takes place out here in the cytoplasm. The rest of cellular respiration takes place in the mitochondria. So glycolysis in the cytoplasm. This is what you got to remember. These are the four things you got to remember for glycolysis. Don't worry about anything else on glycolysis. Takes place in the cytoplasm, uses substrate level phosphorylation. It is anaerobic, which means it does not require oxygen. And the end result of glycolysis is to give you 2 molecules of pyruvic acid. Now, let's show you some, let me explain why that is important. Glycolysis, glucose is a 6-carbon molecule. 1, 2, 3, 4, 5, 6. Pyruvic acid is a 3-carbon molecule. So you take the 6-carbon glucose and split it in half, and that gives you 2 molecules of pyruvic acid. One more bullet that we probably need to see, ATP. You make 4 ATP out of this. So 4 ATP. So what do you need to remember from glycolysis? In the cytoplasm, substrate level phosphorylation, anaerobic, 2 pyruvic acids, and 4 ATP. Okay, now let's go on to the next slide here. Yeah. The final products of glycolysis: 2 pyruvic acids, 2 NADHs, 4 ATPs. Because you've stripped off electrons and hydrogens from the glucose. To make the pyruvic acid, you have to strip off the hydrogens and the oxygens. You make 2 NADHs. You make 4 ATPs. This slide should go along with the previous slide. You make 2 pyruvic acids, the 3-carbon compounds. You strip off enough electrons and hydrogens to make 2 NADHs. You make 4 ATPs, but you ended up with a net gain of 2 ATPs because you have to start the process rolling by releasing the energy in 2 other ATPs. So you burned 2 to make 4, you have a net gain of 2. The most important thing out of glycolysis, besides pyruvic acid, is the fact that you've got, besides the pyruvic acid, you got the 2 pyruvic acids here. This isn't important. What's very important, though, is that you have 2 high energy electrons in each NADH. So you have 4 altogether. You have parked some high energy electrons there. That's what's important. Pyruvic acid and the high energy electrons. 4 ATP is nothing. Cellular respiration, though, takes place in the rest of it takes place in the mitochondria. Here's where we use oxygen. Now, we need ATP. We need to make a lot of ATP. Right now, we were making ATP through substrate level phosphorylation using the enzymes. But we can't store ATP. It only lasts for 7 seconds, and then it breaks down. So we got to keep making it and making it and making it and making it. We can't store it very long. Now we can make ATP. There's 2 ways of making ATP. It's phosphorylation, either substrate level phosphorylation, adding a phosphate to ADP, or oxidative phosphorylation, where we're using a pump in the membrane of the mitochondria. So there's our ATP. The energy is contained in this phosphate bond, this high energy bond right here, when the 3rd phosphate is forced on. That's a high energy bond that will last for about 7 seconds. Okay. Now, we move into the mitochondria. We have the Krebs cycle taking place in the mitochondria. Pyruvic acid the 2 3-carbon compounds come into the mitochondria if we have oxygen present. Pyruvic acid is moved into the mitochondria. And the pyruvic acid enters into what's known as the Krebs cycle. When we get into the Krebs cycle, the pyruvic acid is oxidized and broken down to a 2-carbon compound. That 3rd carbon comes off as carbon dioxide. The 2-carbon compound enters the Krebs cycle proper. Here comes the pyruvic acid right there. There's the 3-carbon compound. It is oxidized here. It gives off a CO2. When it's oxidized, it forms 2 more NADHs. So we've gained 2 more high energy electrons. We end up with a 2-carbon compound that we form acetyl-CoA, and the acetyl-CoA enters the Krebs cycle, and we generate oxaloacetic acid. I know this is getting really confusing. So let's get to the bottom line here. Here's what happens. Pyruvic comes in, it's oxidized, it forms a carbon dioxide. There goes your first carbon dioxide. The pyruvate joins and forms a larger molecule, and we get a 2nd carbon dioxide, and we get a 3rd carbon dioxide. Look at the NADHs forming over here with their little high energy electrons. Now, and this is the Krebs cycle right here. What do you need to remember about the Krebs cycle? Well, it takes place in the mitochondria. It is the source of the high energy electrons. You want to know what the bottom line is? This. The Krebs cycle will generate 2 ATPs, 6 CO2s, 2 FADHs, 6 NADHs, and altogether 8 NADHs. Didn't make a lot of ATP. So far, if you're keeping score, we made 2 ATP net in glycolysis and 2 from the Krebs cycle, but we got a whole boatload of NADHs. Each one of these NADHs has high energy electrons, high energy electrons, high energy electrons. These are the high energy electrons. And that's what we want. We don't care about the ATPs. We've only made a net total of 4 ATPs here. There's a whole bunch of energy available to us, like 280 kilocalories of energy. And what have we got? About 28 kilocalories so far? That's nothing. We've also generated carbon dioxide. Our carbon dioxide comes from the Krebs cycle. That's the only place in our bodies where only process that generates carbon dioxide is the Krebs cycle. So now, this is your bottom line slide on the Krebs cycle. Electron transport consists of 3 enzymes embedded in the membrane of the mitochondria. These NADHs are going to bring hydrogens and electrons to the electron transport chain. 3 different enzymes embedded here, plus what we call the ATP synthase. Oops, didn't even do that. There's our enzyme 1, enzyme 2, and enzyme 3. These are high energy electrons here being delivered along with the hydrogens being delivered. There's high energy electrons and hydrogens coming together. These are low energy hydrogens here. Now, let's go on. Let's get up this and stop. That's why I'm doing this. There we go. Now, what will happen is that in the electron transport chain, we will bring hydrogens and electrons to enzyme 1. The hydrogens will get pumped into the inner membrane matrix here. The energy to pump the hydrogens across the membrane is coming from the high energy electrons. we keep pumping hydrogens across the membrane here. They keep coming back through this structure called the ATP synthase. Every time it goes around, we make an ATP. So we create a concentration gradient high in hydrogens up here. We use the electrons that we've captured from the glucose to pump the hydrogens across the membrane. But they want to get back down. They come back down through the ATP synthase. The hydrogen atoms give up their the hydrogens and the electrons finally break their friendship. The electrons are used to power the hydrogens across the membrane into the space between the inner and outer membrane of the mitochondria. And it looks something like this. Here's our electron transport chain. Here comes NADH carrying electrons and hydrogens. The hydrogens get pumped across using the electrons as power. It maintains a high level of hydrogens here. The electrons are passed to the second part of the chain here. And these hydrogens will eventually come back through the ATP synthase. Once these electrons have given up their energy, they are released back into the matrix of the mitochondria. Those electrons are grabbed by oxygens. The hydrogens that have come through here combine with those electrons to form water. We're busy making ATP here, but we're also making water, lots of water. We've also used the Krebs cycle to release carbon dioxide. And so there's our process. The NADHs carrying the hydrogens and the high energy electrons pump the hydrogens across the membrane there and there and there. And they come back through the ATP synthase. It spins around and produces lots of ATP. And we get about 32 ATP here doing this. We get the bulk of our ATP from the ATP synthase. The hydrogens, when they come through, combine with the low energy electrons, and they're grabbed by the oxygen that's available. This is where oxygen finally works for us at the level of the membrane. There's our oxygen shows up, grabs hydrogens, grabs electrons, there's an oxygen atom grabbing water and hydrogens and electrons. The oxygen's happy, the electrons are happy, the hydrogens are happy. Without oxygen, we don't grab electrons. Without electrons, we don't grab hydrogens. As hydrogens accumulate, and we don't make any more ATP. So we have to have oxygen to make ATP, lots of ATP. And of course, there's our ATP synthase. Every time this thing, it literally looks like a rotor. And every time it goes around, it makes an ATP mechanically. So that's what it looks like. Now, last terms here. Glycogenesis, the creation of glycogen when we have too much glucose. We store this in the liver and in muscle. Glycogenolysis, the breakdown of glycogen to get glucose when we're low in glucose. And gluconeogenesis, the creation of new glucose. Genesis means creation, neo means new, the creation of new glucose from fats and from proteins when we desperately need glucose. Now, what happens if we don't make pyruvic acid? We make lactic acid. Lactic acid is what causes our muscles to hurt when we exercise. So, okay, and that is the end of metabolism. A couple of things that are important. The end result of glycolysis, I showed you that. The end result of the Krebs cycle, and what does the oxygen do for us? So, anyway, let me get out of the sharing. Any questions on any of that? Okay. You have a question? shoot me an email and I'll be happy to answer it. So if there are no questions, I'm going to get us out of here and I will see you after break.